So thank you for being here today. This is going to be our agenda for today's program. We're going to start with a brief introduction to the Allen Institute and where to find our resources for educators. We'll then do an overview of selected resources available at brain-map.org. That's the main portal for all of our open resources from the Allen Institute for Brain Science and some of the professionals behind that work. And then I will walk you through an interactive demonstration of the lesson and virtual experiment, the building blocks of the brain. And then we'll have plenty of time for question and answer at the end. So if you have questions, go ahead and use the Q&A button down at the bottom of the screen and enter those questions there. We'll be keeping an eye on that Q&A panel during the presentation, and we'll answer questions either as they come up or we'll save most of them to the end. Again, please make sure you're sending all of your chat messages to all panelists and attendees so other people can see your message. If your Zoom name isn't your actual name, please send a private message to moderator Christina, or she will be reaching out to you to get yourself renamed. If you're requesting Washington State Teachers Continuing Education Clock Hours, full instructions on how to do that are at the link sent in your webinar registration confirmation on the page you used to log in today. So a little bit about us. Luke and I are going to be your presenters today. Luke is the Director for Scientific Operations at the Allen Institute for Brain Science, where he's been for about 3 years. And I'm the Training and Outreach Specialist for the Allen Institute, where I've been for about 2 years. We'll both introduce ourselves further at the end of our presentations. So a little bit about the Allen Institute. We're a biological sciences nonprofit located in Seattle, Washington. We have focused research areas in neuroscience, cell biology, and immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across our institutes, our scientific focus is on tackling complex, broad, and hard problems in the fundamental sciences. And our approach is guided by 3 principles: big, team, and open science, which I'll return to in a moment. And our work generates data, knowledge, and analysis tools, all of which we share publicly. And to date, we've collected about 8 petabytes of data, which is about 1.7 million DVDs worth. So our 3 core principles are team science, big science, and open science. The most relevant one for today's presentation is open science, which is that we share all of our data for you to use in your research or with your students. We're going to be focusing today on resources that are found at brain-map.org. But we also have resources for cell biology at ellencell.org. So if any of you are teaching cell biology or do research in cell biology, you should check that out as well. So the 2 places to get started for educators are first, alleninstitute.org/learn, which you probably visited when you were registering for this webinar. It includes lesson plans, posters for when you're back in your classroom, and resources for teaching neuroscience and cell biology. We also have some more resources that will be coming soon. allencell.org/educational-resources has links to cell biology specific data resources for if you also teach general biology or want to pass it on to your colleagues. The previous 2 webinars in this series covered lessons and data from allencell.org, and you can find the recordings of those webinars, and you'll find the recording of this one on the page where you registered for this, which is also linked from alleninstitute.org/learn. So we're going to be talking about the building blocks of the brain, our featured lesson for today. You can find that, again, by going to alleninstitute.org/learn and scroll down till you find this picture of neurons that you can see right here. Or it's linked on the page that you used to join this webinar as well. So most of our resources were developed with research applications in mind, but they can be used in classrooms to provide students with a genuine research experience without the need for access to lab facilities that might not be available in your school, and when you're at home and you're distance learning and you don't have access to a lab at all. We know that high-speed internet is far from universal, but for many students who are currently learning at home, these virtual experiments can provide a bridge for continuing their science education when you can't get into a lab in person. And even when you are back in your classrooms, we hope our open data can help you study human cells and the brain by providing access to types of data that typical high school and college labs don't necessarily have the facilities or the time to collect. So I'm going to hand it over to Luke. Again, if you haven't already done so, please send Christina a message to have yourself renamed if you need. And many thanks to Christina for helping out with the questions and the chat. She'll be answering some of those questions for us. We do encourage you to chat with each other as we go through these presentations, but questions should go in the Q&A. So Luke, I will hand it over to you.
Okay, thank you. Having a little trouble sharing here, but we'll get there. Okay. Are folks seeing the screen?
We are.
All right, excellent. Thank you for the wonderful introduction. I'm Luke Esposito, Director of Scientific Operations at the Allen Institute for Brain Science, and I'm going to present to you an introduction to and some of the scientific background on both the Allen Human Brain Atlas and the Allen Cell Types Database. So we know that our audience consists of both researchers and educators, and we aim to connect with all members of our audience. We know that researchers include current or new users of our data and website at brain-map.org, basic science users, as well as those engaged in more disease-focused, translational, and in some case, clinical users. We also know that computational biologists are big users of our data and our resources. We know that educators include secondary educators who might be introducing neuroscience to their students for the first time, as well as undergraduate educators who might be looking for course-based research experiences or expanded data sources for student-independent research projects. We also know that graduate educators use our tools to demonstrate to their students what's done by their peers in the field. And we know in many cases, it's some combination thereof, which likely includes many of you here today. So as Caitlin mentioned, open science is a real key component of our mission. This means that we share our data online as soon as it passes quality control and generate user-friendly resources to showcase the data. This is just a brief overview of the roughly 17-year history of the Allen Institute for Brain Science, highlighting some of the resources that are available for free on our website. We won't try to cover all of these, but we'll hit just a few high notes. So in September 2003, the Institute was launched, and this came on the heels of the completion of the mouse genome project in 2002. So for the first time in the history of science, we had a complete list of all of the genes from the mouse genome that could be expressed at any time in any tissue. So this begged the question, where in the mouse brain, in the young adult mouse brain, are these genes expressed? So the first project was launched, the Allen Mouse Brain Atlas, and just over a year after the Institute was launched, the first iteration of that tool was released in December 2004. That came at the same, 2003 was also the time that the Human Genome Project was completed. So that begged the question, where in the brain, where in the adult brain are all of the human genes potentially expressed? And that is the result. The result of that is the Allen Human Brain Atlas, which was first released in May 2010 to answer that precise question, where in the brain are the genes expressed? And that will be the subject of the first part of the talk. In 2015, the Institute launched the first iteration of the Allen Cell Types Database. This is done using single cell work that we will talk about in the second half of the presentation. And the most recent addition to this tool is this Transcriptomics Explorer that you'll hear a little bit about in Caitlin's lesson plan. In addition to these open access resources, we publish extensively in the scientific literature, and we also create open science research tools that include software and biomaterials such as transgenic mice and recombinant viruses. So the presentation is broken down into 3 sections. In the first part, we'll talk about the Allen Human Brain Atlas, and then we'll get into this concept of cell types. So what are they and why are they important? And we'll give you some insight into how we approach cell type discovery at the Institute. So this is a cross section of a human brain on a large glass slide. This is a coronal section, and you can see that this brain is indeed big, especially when compared to a mouse brain. This is also, as we all know, a very complex tissue, and one cannot do complex or detailed gene expression analysis on a live human being. So this requires post-mortem tissue, which is how this study was approached. The authors took an all genes, all structure approach. And to do that, they started with these thick sections from post-mortem brain. There were a total of six subjects who donated their brain to the study. They took from these thick sections, thin sections that they could use for histological stains to identify anatomical landmarks. Based on those anatomical landmarks, they then did dissections, and in some cases, microdissections with the help of laser capture microdissection to isolate individual brain structures. They placed those brain structures in individual tubes, extracted the RNA, and then looked at gene expression levels using the DNA microarray technology that was state-of-the-art at this time. From those individual results from each of those individual tubes, the gene expression was put back into three-dimensional context by basically stitching the brain back together using the anatomical map that was drawn from the adjacent sections. This was a large data set. This is a large data set. This is approximately 700 structures and around 24,000 genes per structure. This generates a very large data set, and I think large data sets can sometimes be intimidating, particularly to a student who might be new to looking at large data sets for the first time. So one way to approach large data sets is starting by asking a simple question. And there are many types of questions that you can use using this atlas and this resource. For example, you can ask an anatomy-focused question. So which genes are expressed in my brain region or regions of interest? You can ask a gene-focused question. Which brain regions, or structures as we call them, express my gene or genes of interest? You can also take a correlation-focused approach where you look at more than one gene to see which genes are co-expressed along with the gene or genes of interest. You can also take a category-focused approach. So you can ask a question, how do genes that converge on a biological pathway or disease process correlate with one another? And for this purpose, we've provided several pre-curated lists of genes for this purpose. One can also ask species-focused questions. So how does the expression pattern of my gene or genes of interest correlate between human and mouse? Before we show you the results and the data from this tool, just a couple of technical notes. First of all, by default, expression levels are expressed relative to other regions across the whole brain. And for this purpose, we use Z-score analysis that Caitlin will touch on in her lesson plan. This is also worth noting that this is bulk tissue expression data. This is multiple cells per brain structure. It's not single cell work, which we will focus on in the second half of the talk, but it's the high anatomic resolution and the completeness of this assessment that is the real strength of this Human Brain Atlas tool. Okay, so how does one get started? So you go to brain-map.org. There's no need to register. There's no hurdles there. You just jump right on. You click on the Human Brain Atlas, View Atlas, and you come up with a landing page that looks something like this. By default, you can select from this gene search, which is in this radio button shown here, sort of on the upper left of that panel. And there are other search options that we will get into a little bit more as we go through. So this is one place to start. I like to start simple. I like to ask a question that I already know something about the answer. This helps me calibrate the response that I get from the tool. It helps me understand how informative this tool is going to be for my purposes. So for me, I would start with a gene called SNCA, which codes for a protein called alpha-synuclein. I worked on this protein for several years when I was working in industry. This is a Parkinson's disease-related gene. It's widely expressed, and the protein is very abundant, present at high levels in the cytosol, as well as associated with membranes. This protein has a tendency to misfold and aggregate, and it's this active gain of function that causes neurodegeneration. And indeed, familial mutations in the gene increase propensity for misfolding and aggregation, which make this protein a very attractive and widely studied therapeutic target. So how do we start? So this is just a fraction of that window. that we started with at the beginning. This is the Gene Search Radio tab. Clicked on the left. I typed in SNCA and hit search, and I get a result that looks something like this. And this is a heat map, which contains a lot of data. So we'll walk you through this so that you can get the feel for it and then be able to use it to your needs. So each row is a gene probe corresponding to the alpha-synuclein gene. So here there are 2 probes corresponding to this gene. In the atlas, roughly 93% of all of the genes have more than 1 probe associated with them. Each column is a combination of the donor and an individual brain structure from that donor. As I mentioned earlier, there were 6 donors that were part of this study. Here we're looking at mostly 1 donor, which is this orange band across the top. And for each structure in the brain, there's an individual band, which corresponds again to that individual and that individual brain structure. On these heat maps, red equals hot or high expression, green equals cool or low expression. And so you can see here just at a glance that alpha-synuclein is expressed in many regions of the cortex at relatively high levels. For example, the temporal pole of the cerebral cortex, which is that single band that I pointed to here on the left. And you can also see that it's expressed at relatively lower levels in regions of the thalamus, including this medial geniculate complex, which is part of the dorsal thalamus. I said at the beginning of this section on alpha-synuclein that this is a widely expressed gene, and that's true. The view that we looked at by default focuses on differences between regions, but if you wish to look at something which is a closer approximation to absolute expression levels, you can go to this color map toggle and choose log2 intensity, and that will reveal overall expression for alpha-synuclein is indeed very high, as indicated by lots of red in most, but not all of these regions. And so, as I said, this de-emphasizes expression between brain regions or brain structures, but it shows that this gene is indeed a very highly expressed gene. So again, this is calibrating to my expectation, showing that this result is showing me basically what I expect and giving me insight into how to exercise the different options that the tool provides. Another question is, what is the expression across the brain of different Alzheimer's disease related genes? And this would be a classification search. An example of that is shown here, where under this gene classification drop-down menu that I've circled here in red, you can choose from several curated lists of genes. Here I've chosen Alzheimer's disease, which is a list of genes that were known at the time that this tool was generated to be related to Alzheimer's disease. The results are at the bottom, and you can see there's a lot of data here. Again, each row is a gene probe, and in this case, most of the genes shown have more than 1 probe. And then each column is an individual band corresponding to an individual structure from an individual donor. And here the data is more compressed, so you can see all 6 donors on the same screen. This is obviously a lot of data, and so we're not going to try to dig into this and unpack all of this data here, but it's worth saying that this is really a good place for a student to start. They can maybe use some information that they already have, some information or insight they might have about Alzheimer's disease, and then use that as a jumping off point for asking questions and maybe forming some hypotheses and then testing those hypotheses with that data. Caitlin will show you an anatomical view of gene expression in the lesson plan, so I wanted to touch on that here. So this is basically looking at 1 gene probe at a time. This is for this angiotensin 1 converting enzyme gene, or ACE, which is from that Alzheimer's disease related gene list. And the results here, this is from 2 subjects, show that on the left, you'll see lots of green and black, indicating that in many of the cortical regions that are shown here, there's relatively low expression of this gene. But if you look at the basal ganglia, including the striatum, which is shown here in the middle, you'll see lots of red, indicating this gene is highly expressed in that region. Okay, that was all work done with bulk tissue analysis. Again, that was a very wide and broad study to look across all anatomical regions. And in the second half of the talk, we're going to focus on work that was done using single cell analysis across a more narrow region of brain, but we will give you some insight into what kind of information this, what we call cell type analysis, can provide. So what is a cell type? Why are they important? And then we'll finish with an overview of how we approach cell type discovery at the Allen Institute. Okay, so what is this concept of cell types? This is a population-based classification, which is dependent upon some discontinuous variation within the population. A cell that is a member of a group or a type is more like others within that group than it is cells outside that group. Historically, neurons have been described based on their anatomical location and their morphology or shape. And this is work that goes back more than 100 years to the time of Ramon y Cajal, where he generated these beautiful pictures of the different shapes of neurons in the cortex, the cerebellum, and the retina. Another way to describe neurons would be based on the task that they carry out or their function, although in the complex tissue of the brain, this is often not immediately clear. Another way to classify cells would be to group them based on shared features, which are obtained by one or more measurable criteria. And these features are ideally quantitative and linked to function. Cell type classification is hierarchical with a major class at the top, for example, a glutamatergic excitatory neuron or a GABAergic inhibitory neuron, followed by a subclass, a type, and in some cases, a split into a subtype based on new information. We think cell types should be treated as a testable hypothesis subject to change upon availability of additional new data. So historically, the definitions of cell types have been relatively coarse. They've been really dependent upon anatomical location and morphological descriptions. And in some cases, the dependence upon the availability of marker genes that can be used for immunohistochemical analysis, but there's not very many of these marker genes available. However, recent technical advances enable a much finer characterization and provide new opportunities for cell types research. Those include single cell and single nucleus techniques, next generation DNA sequencing, which is much more efficient and economical, transgenic tools and technology that allow us to access and report on more rare cell types, high resolution imaging and reconstructions that allow beautiful images such as the ones shown here on the right of the slide, as well as electron microscopy that we won't get into today, but is also a source of very high resolution images. And then there's an increase in computational power, statistical methods, and dimensionality reduction algorithms that are useful for really getting a handle on these large high dimensional data sets. And this is really this whole field of data science that I'm sure you and many of your students are familiar with and perhaps actively engaged in. There's also a renewed appreciation in recent years for what we call the descriptive enterprise, which is generating these large descriptive data sets, which can then be used to form hypotheses that can be tested in the lab. And this is evidenced by increased National Institute for Health supported efforts in this regard. Okay, so why do we care about cell types? Well, we think it's obvious. To understand the whole, we must understand the parts. So just like to understand how your cell phone works, you might take it apart and assess the function of its individual components. We think the same is true for the brain. In addition, we know that in neurodegenerative disease, there's often damage or loss of specific cell populations, particularly early in the disease. So we think that if we can better describe these different types of cells in normal brain, we might be able to understand how to compensate for their loss and perhaps generate more effective therapeutics for these largely intractable diseases. But our first goal is to identify and characterize cell types in the healthy brain. So what do we mean by organizing cell types into hierarchical clusters? So imagine you have a population of cells, each of which is represented by an individual dot shown here in this collection of dots on the left. You then apply some measurable criteria to that population, and you group cells based on their similarities or differences. So cells that are similar are assigned a similar color, and cells that are different are assigned a different color. You then use a computer algorithm, a clustering algorithm, to generate a dendrogram, or this tree-like diagram, which is shown here on the right, which illustrates this hierarchy of clusters. So cells that are members of a cluster or a group comprise a type. So they're very similar to one another, and they're different than cells that are in different clusters or different types. It's also worth noting that cells that are in neighboring clusters are more like one another than are cells that are in more distant clusters in the tree. Okay, so that's just an overview, sort of some of the theory and background on cell types. So let's get into a little bit of how we approach cell type discovery at the Allen Institute. So we like to move from individual cells to what we call multidimensional cell types by collecting data using different methods that are relevant to the function of neurons and other cells in the brain, such as non-neuronal cells. So we collect 3 different types of data. We collect transcriptomic data, which describes the gene expression patterns for those single cells, and we use single cell or single nucleus RNA-seq for this purpose. Secondly, we like to understand the morphology or shape of the cells, and that can be done using 3-dimensional reconstruction of biocytin-filled cells, and in some cases, electron microscopy. Thirdly, we wish to define the electrophysiological properties of the cells, and this is done with whole cell patch clamp recordings, and this really defines the language that the neurons use to speak to one another, the firing properties that is really the essence of function. So from these individual data sets, we generate what we call property-specific taxonomies. For transcriptomics, it's a T-type. For morphology, it's an M-type. And for electrophysiology, it's an E-type. We can also carry out what's called joint clustering, in which the algorithm accounts for more than one data modality, and we can generate these multidimensional taxonomies using that joint clustering. Here we're showing the 46 ME types, which takes into account both morphology and electrophysiology, and this was work published in 2019 in Nature Neuroscience. And more recently, we've defined MET types, which also takes into account transcriptomics data, and that work is under peer review, but it's also available already for your viewing on bioRxiv, as well as much of the data is available on our website. So we want to get towards what we can consider a unified cell type taxonomy. And of course, that grows more complex the deeper you go within a brain region, or the wider you go across the brain, or even outside the brain across the whole body. So this is just a relatively simple view of mouse cortex as of roughly 4 years ago, showing a fairly simple organization with a major class at the top. Again, this could be a glutamatergic excitatory neuron or a GABAergic inhibitory neuron, followed by a subclass, which is indicated by a marker gene, and in the case of excitatory neurons, a projection pattern. Ultimately, we can identify types, which include information about anatomy, plus additional information such as marker genes. And in some cases, there's a subtype or a split based on new information. But as I said at the top of the slide, it gets more complex the deeper you go. So one way to really go deep is to use transcriptomics. It's highly dimensional. There are many genes, and you can look at all of the genes across single cells to really do a deep study. And this was done for 2 regions of the mouse adult cortex. The authors looked at 24,000 cells across the primary visual cortex and the anterior lateral motor cortex, and they identified 133 clusters or types. The organization of those types are shown here. Don't worry about all the details. We don't expect you to read all 133 names of the different cell types, but just take away that there are these major classes, including non-neuronal cells, inhibitory neurons, and excitatory neurons, and then these subclasses and ultimately individual types. And this was published in 2018 in Nature if you want to go deeper. A few take-home messages from this work. This is approaching saturation, so it's unlikely that analyzing many more cells will reveal many more cell types, although it is possible that some rare cell types would be identified if we were able to go deeper. As I mentioned, there's this hierarchical organization, and this is pretty much what we expected, although there were indeed new subclasses and types identified, including this LAMP5 subclass of inhibitory neurons. At the biological level, there were some interesting messages. Number one is there's this common inhibitory cell types. In other words, in these two distinct anatomical regions, one in the back of the brain, one in the front, many of the inhibitory cell types were actually the same. They were conserved. In addition, we noted distinct excitatory cell types across these regions. So they were different than inhibitory neurons that were more conserved. And you might expect that for excitatory neurons because they have distinct projection patterns depending on their anatomical location. Okay, so that was work done in mouse. We're very interested, of course, in human. So that mouse study was used as a baseline for a comparative study with cell types in humans. This was work done in human middle temporal gyrus. This is part of the cortex. And these were done on 8 patient samples, 4 of which were postmortem, and 4 of which were surgical resection samples, which were obtained through collaboration with local surgeons under patients who consented to having this piece of resected brain donated to the Institute for use in these studies. These were patients with severe forms of epilepsy, and in one case, a tumor resection as well as epilepsy. So this was single nucleus RNA-seq. We showed in a separate study that the results between single nuclei and single cells are indeed comparable. And so there were some really interesting messages and some findings in this study. So the authors noted cell type homology, significant cell type homology between mouse and human, despite 96 million years of divergence and about a 1,200-fold difference in cortical size. So I think this is very striking. There's this conservation of cell types across this wide evolutionary space. They identified these conserved cell types as consensus cell types that were shown to be similar across species. And they concluded that the overall organization is very similar, but indeed, as you would expect, the details indeed differ. So we'll talk about that. So for example, they showed that there was divergence of gene expression across species within a cell type, as well as an anatomical or laminar shift of homologous cell types across species. They showed that gene classes that best differentiate cell types within a species show the most divergent expression pattern between species. And a notable example of that is this serotonin receptors family, which diverged greatly between species, which creates challenges for mouse models for neuropsychiatric disorders that involve serotonin signaling. They concluded that there's a basic transcriptomic similarity of cell types, punctuated by differences in proportions, location, and gene expression between species, which may greatly influence microcircuit function, which is why it has been said you cannot have dinner conversation with a mouse. Okay, so that concludes the scientific portion of this talk. As Caitlin mentioned at the top, we thought it would be helpful to describe our career paths and give you an idea of where we started and what we've done. So I grew up in a small city in upstate New York called Utica. I'm the son of a high school art teacher and school principal, so I can really appreciate all the great work educators do. I did my undergraduate work at Boston College, where I purified proteins that were then used for X-ray crystallography studies. I obtained my PhD at Emory University in Atlanta in biochemistry and molecular biology. I focused on how mitochondrial DNA mutations affect mitochondrial bioenergetics and the redox state of the cell. This, of course, has implications for different theories of aging as well as disease. And we know that many of the rare mitochondrial diseases that result from mitochondrial DNA mutations result in neurological phenotypes. So that got me really interested in neuroscience. I knew that I wanted to dedicate the rest of my career to neuroscience. And to do that, I wanted to study a more common neurodegenerative disease. And we know that the most common disease is Alzheimer's disease. So I moved to University of California, San Francisco, specifically the Gladstone Institute of Neurological Disease. And I embarked on my postdoctoral fellowship to assess the effect of oxidative stress on misfolded proteins and how together these two things would exacerbate Alzheimer's disease-like phenotypes in transgenic mice. Following my postdoc, I was recruited to a company in Seattle, originally called Proteotech, ultimately called Protomed, where I served in several roles, including Director of Neuroscience Research and Vice President for Product Development. Our focus was on drug discovery targeting misfolded proteins, for example, alpha-synuclein in Parkinson's disease, and the tau protein and amyloid beta peptide, which are involved in Alzheimer's disease. We also studied rare diseases that result from misfolded proteins, and these affect tissues in the periphery, such as the heart, liver, and kidney. I joined the Allen Institute in May of 2017, and I would say that my job here is really at the scientific and business objective interface. So where these two things meet is where I sit. I would classify my job as a scientific management position. So it has elements of governance, scientific evaluation, and strategy and planning of our scientific projects and goals. At the practical level, my team and myself are really here to ensure that we have synergy across our different functional units. And so let's talk a little bit about some of the career tracks, both in my group and outside my group at the Institute. So we have research associates. These are folks who typically hold a bachelor's or a master's in a science, typically a life science, and they play a real critical role in data collection. In some cases, they also contribute to analysis, and they may manage other research associates, so have people manager roles. We also have scientific program and project managers. Typically, these folks have a PhD in life science, and they play a real key role in project and budget planning, including grant applications, both at the application stage as well as the execution stage after the award has been made. And their major goal is to ensure that project milestones are met. And we're a very milestone-driven organization. We also have project coordinators. These people typically have a bachelor's or a master's degree in a science. Many of them have a bent towards technology, and they play a key role in cross-team coordination, standardization of methods such as standard operating procedures, many of which we now make available for free at protocols.io, as well as quality control, which is a key component of everything we do. And they're always looking for ways to improve processes in the name of operational efficiency. We also have science support teams. Depending on the role, they may have either a high school diploma, an associate's degree, or a bachelor's in science. And their major mission is to provide care, training, and other critical services for our transgenic mice. And then finally, the job family of scientists. These people have a PhD in the life science, physics, math, computer, or data science, and they're involved in study design, data collection, analysis of that data, and ultimately dissemination of that information to the scientific community through manuscripts and presentations at scientific meetings. And they're really key components of the engine that drives discovery at the Institute. So I will pause there and hand it back to Caitlin.
All right. So can you see my screen?
Yes.
Great. So now we're going to talk about the featured lesson for this webinar. Again, this lesson includes worksheets, a virtual lab, and instructions for how to collect the data, and it uses those two databases that we just covered, the Allen Human Brain Atlas and the Allen Cell Types database. This lesson is linked from the registration page that you received to log into the webinar, or go to alleninstitute.org and scroll down, click on this lesson. The lesson is intended to be easy to modify according to the level of your students and the needs of your class. You might just do the lab part, skip the worksheets. You might just do the worksheets, just poke around at the data, but not really collect anything. Or you might modify the data collection to more closely align with another experiment in your curriculum. But the idea is that the materials are intended to give you a starting point, because it's always easier to edit than to create from scratch. So one of the core goals of this lesson as written is to expose your students to some current research in neuroscience and some types of data that are commonly encountered in the field. We want them to understand how much we still don't know about the brain, that there are opportunities to move the field forward, that this isn't something that's just done and sewn up in a textbook. So students are going to be able to describe some of those key open questions in neuroscience, learn about gene expression in the brain and how the function of a brain region is related to that, learn about some differences between studying brain regions in single cells, and starting to think about key features of individual neurons that can be used to sort cells into types. Students are also going to collect and interpret a type of gene expression data, that's from the Human Brain Atlas, and collect some basic electrophysiology and morphology data from some human brain cells. And they're going to be asked to design their own experiment that uses the same type of data that they use in the lesson. So they'll be able to apply their knowledge. So the lesson is written. All of these pieces are flexible. It consists of a pre-lesson homework, first class session with a discussion, a pre-lab homework. This could also be completed in class. Second class session would be where they would collect that data in the virtual lab. And then they do a little lab report, analyze the data that they collected, and answer some guided reflection questions. You can do all of this as homework. You can do it all in class. You can modify and replace sections. But I'm going to go through it as written. So while working at school, a computer lab plus paper or digital worksheets would be a really great way to run this exercise. And these worksheets were designed to be printable. But while at home, students might have access to a wider range of devices, and that's going to impact how this lab can be run. If your students only have access to a phone, we would recommend modifying this lesson to concentrate on just the worksheets, reading, and discussion, and skip the data collection. As you can see from this photo, there's just not enough screen real estate on a phone to be able to use the data collection or to do the data collection on our website. The website was designed for a computer, but it will work on a touch screen. So if your students have access to a tablet, that will work just fine. It's not a touchscreen phone that's the problem, it's that they need a little bit more space. And we also recommend, especially for high school students, that they work in groups to collect that data. If they haven't seen this type of data before, that's really going to help them. So for that pre-lesson assignment, if you're following along with the lesson plan PDF, this is going to be starting on page 11 is where the student worksheets begin. They're going to read 2 articles on our website. 5 Unsolved Mysteries of the Brain is an article that covers some major open questions in the field of neuroscience. And then students will be prompted to think about what are they surprised about in the article and which of those open questions is most intriguing to them. And then the article, Genes that Build Our Brains and May Drive Neuropsychiatric Diseases, talks about applications of genetic data from a healthy brain to also studying neuropsychiatric diseases. And they're going to answer some more questions about that as well. And then we have a few more discussion questions that were designed for working in class. You can do these in person, virtual, synchronously, or asking them to just work asynchronously together, or working alone. This is just going to depend on the needs of your class, and you know that far better than we do. And then we have a pre-lab worksheet assignment. This is designed to be completed between the class discussion and before running the lab. And it's pretty short. You could also do it during a class session with those discussion questions. The main things here are for students to read the experimental protocol and anticipate what they're going to need to do to complete each step and where they might run into trouble. They're also going to read a short animated explainer of how cells are categorized into types based on their gene expression. Now, they don't explore the single cell transcriptomics data as described in this article. They explore related genetic data that isn't single cell, and they explore related single cell data from similar cells, but they only look at the electrophysiology and morphology. But this essay does a really nice job of explaining why cell types are useful for scientists to understand. At the time that this lesson was produced, we didn't have these beautiful online data visualizations of single cell transcriptomics that we do now, but we're working on a lesson addendum that uses it. Or you can just have your students check out some of that data as an added challenge. So stay tuned for that add-on. We're going to be releasing that hopefully soon. And these transcriptomics viewers, I will show you how to find them. All right, so now we're going to walk through the actual data collection, the virtual lab part. So students are going to learn how to collect data from our open databases on our website, which will prime them to start conducting their own experiments using our data once they're familiar with what's available and how to access it. So it's a 3-step data collection process. Part 1 is going to ask students to select some genes of interest and regions of interest that they'll then use to collect data from our human brain atlas. They're also going to look up the functions of those genes using a gene database. In part 2, they're going to look at the relative gene expression of their genes of interest in our database. In that post-lab worksheet, they're going to interpret the data that they collected, which is in the form of z-scores. So in this section, they're just collecting the data, they'll interpret it afterwards. And then part 3 will ask students to explore some data on single neurons. They're going to look at the electrophysiology of a few single cells and their shape. So let's get to the data collection. So for the first part of the virtual lab, students are going to get set up with some genes and brain regions. The worksheet gives 5 sample genes for them to work with, but they can actually pick any gene. The microarray data in our human brain atlas includes the full human genome. We also picked a specific probe for each gene. Almost all of the genes have more than one probe. If they pick other genes, they can select any of the probes. You can read more about how the probes work and the documentation on our data page. They're then going to visit the NIH Gene Explorer to look up the protein associated with their genes and their general function. You can use another gene database if you prefer. So this student has selected 3 genes, DRD1, NPY1R, and SCN1A. And then they're going to go visit the NIH gene database. So let's do DRD1. And we'll give that a moment to load. So that's DRD1, dopamine receptor D1. And this is going to take us to a page that describes the function of this gene. This database is linked from the lesson plan. You can, again, use any gene database that you prefer. So our student has now looked up the function of these 3 genes, specifically what protein do they express, or do they lead to the expression of. Great, so this student now has some genes, and it's time to go look at their expression patterns. So we're going to head over to brain-map.org. So that's brain-map.org, and then you're gonna scroll down to the Human Brain Atlas. And then you're going to use this search box here to search for your gene of interest. So we're going to do the same gene. And you can see this gene has a bunch of probes that were used for it. We're just going to select the one probe we want. And again, this is that heat map that Luke showed you. So we have the subject, or the donors are listed along the top here. The brain regions are this second layer of colored bars. And then this heat map, red means more expression, green means less expression. The heat map is great for comparing across genes or probes at a glance, but it doesn't give us much quantitative detail or a sense of the anatomy. So we're just going to go in and take a closer look at the anatomy for this gene. So if you go in, it shows the heat maps of the relative expression for each gene for each individual. It's only going to show one gene at a time, but it puts in the context of the anatomy. There's 6 individuals here, and you can see that their expression patterns are not identical. The heat map is going to show the relative expression level for that gene across the whole brain in the form of a z-score, and that z-score is calculated across only that gene. And it basically tells you the relative expression level for that region. So, unsurprisingly, we've got high expression of DRD1 in these basal ganglia regions. So your students are going to find their regions of interest, and they're going to record the z-score for that region of interest for that gene. So this student has now collected their data from 3 brain regions. We asked them to collect the data from at least 3 brains. This student has collected the data from 4. Again, this data is in the form of z-scores.
Hey, Caitlin,
yes?
Could you re-show, someone's requesting, could you show how to get from heat map to anatomy one more time?
Yes, I absolutely can. So I've just gone back to the home page of the Human Brain Atlas. So again, you get to that by going to the home page of Allen Brain Map. You can hover over the top here where it says Atlases and Data, select Human Brain Atlas and Microarray Data, or you can scroll down here to Human Brain Atlas. And then right here where it says gene name, you're going to enter the name of the gene that you're searching for. So I was doing DRD1. And then you select that gene. Before I go to show you how to get to that anatomical view, I'm also going to just quick point out this documentation button is where you can go to find much more methodological detail on how all of this data was collected. including the link that I dropped in the Q&A earlier, this is where you would get to that. So again, you just click anywhere on that heat map for your probe of choice. And then up here where it says gene info, just click on the gene symbol. And that's going to take you here to that heat map. Great. So this student has now collected their data from one brain region or one gene for 3 brain regions, and it looks more or less the same to repeat that for the other 2 genes. So now for part 3, we're going to move to a different one of our databases. This is going to go to the Allen Cell Types database. So if you go up here, click anywhere that it says Allen Brain Map or Allen Brain Atlas, and it will take you back to our home page. The students are going to go into our cell types database. This isn't going to be an apples to apples or even an apples to oranges genetic comparison here. It's more like apples to carrots. The takeaway in this section of the lesson is to start introducing a few ideas, studying the brain at different scales, cellular versus regional, and the idea of cell types. And we'll be adding the transcriptomics addendum to this lesson. We'll fit in right here. So I've gotten to the cell types homepage. Again, if you want to know more about the methods used to collect this data, click the documentation link on any page. And then we're going to go to cell feature search right here in the top bar. In this particular application, we're interested in looking at human data. So I'll select human. And you don't have to look at only cells that have a 3D reconstruction of their morphology, but for right now, that's what I'm going to show you. So I've selected over here, reconstruction type full. And then students can use this dropdown to select cells that are in their regions of interest that they selected in the previous part of the lab. And if you scroll down below, you can see each of these tiles represents data from one single cell, and these are all human cells in this instance. And if you click on electrophysiology, it's going to open a new window that loads the data. So it gives a little summary up here of the electrophysiology of this cell. And the students are going to collect the firing rate and the resting potential as part of this particular lab exercise. But I would also suggest that you direct them to look down here where they can see the stimuli that we applied to the cell, that's this graph, and the cell's response. So you can look at what happens in response to each of these different stimuli. And you can see the cell's response to that. I'll also mention that we recently released data that we call patch-seq data. That includes morphology, electrophysiology, and single-cell transcriptomics, all from the single cell. But just so you know, that doesn't appear in this viewer. It's in a different spot on our website, and we'll have an online viewer available for that soon. It doesn't have one right now. The RNA-seq button at the top of the screen here will take you to those transcriptomics viewers, and we'll come back to those in a moment. So now I'm going to show you the morphology of this cell. So if you scroll back up to the top and see where it says View Morphology, that will open yet another tab. And you can see this is what that cell looks like in the dish. It's been stained, so you can see its whole shape, and that a 3D reconstruction is available here. So we're asking students to take a look at that and sketch it as best they can. It's also been color-coded to show like axon versus dendrite. At the bottom of each of these pages, both electrophysiology and morphology, there are buttons that allow you to download the data for that cell. Download morphology here. Data available as an XML to download the electrophysiology data. And if you go back to this cell homepage that has all of the cells, you can download all of that. So if you want to use this for research purposes, you can download the data in bulk. And that's true for the human brain atlas as well. So our student has now collected data for one brain region, MFG, for 3 cells. The data is going to look, again, more or less the same for the other 2 regions. And they can collect data for more than 3 cells if they're available. They're going to calculate some basic statistics, but the sample sizes are going to be small. There's not that many cells available in this data set. If you're teaching a more general biology class than neuroscience specifically, you can actually check out our cell biology lesson plan, mitosis and microscopy, and that one actually gets in a little bit more deeply into sample size effects. It asks students to collect data individually and then as a class and look at differences comparing the different sample sizes. And then everything from here out in this lesson plan is going to be analysis. Before I move on to that, I'm going to show you the new transcriptomics viewers for our single cell gene expression data. These are relatively new. They came out in June 2020, the current versions. And you can find those right now. They're listed in the new data and tools, or you can go to cell types database and then RNA-seq data. That was right here, RNA-seq data. And then you can click explore and analyze on each of these individual data sets to look at their transcriptomics explorer. So I'm picking one of the human data sets. We'll give it a moment to load. So what you're looking at here is the transcriptomic profile. So we've got a bunch of genes here for cells that have been pre-sorted based on their cell type. So we've got inhibitory L1-2, PAC6 SCGN type here. And you're looking at a few key genes will pop up automatically. You can also add any other genes that you want up here in this add genes box. We've just pre-populated ones that contribute a lot. So stay tuned for that addendum to this lesson plan that uses this transcriptomics viewer. So the rest of this lesson is going to be data analysis and reflection questions. The students are going to interpret the gene expression patterns from the Human Brain Atlas and link the gene expression patterns to the function of the proteins and start thinking about how that relates to the function of the region. Starting to understand the difference between studying a region of a brain and a structure versus individual cells, and applying their knowledge from healthy brains to the study of disease. They're also going to pick up specific skills in interpreting Z-scores, and they're going to be asked to design their own experiment. Start thinking about how they would apply the data that they collected. We also have some above and beyond questions. These are additional questions for highly motivated or advanced students, potentially extra credit. These are a little more sophisticated scientifically. They're going to ask about study design and cell typing, and they're going to have a few questions are computer science conceptual. They don't require any coding. Students who are interested in pursuing independent projects using our databases should start here. It might help inspire them some research ideas. Students can then continue exploring our data and our open source software. We use mostly Python and R for our analysis. So if you are looking to teach computer science for life sciences, you can start diving into that and use our data as the subject of analysis. So what's next for students after this? Independent research projects. You can combine our data with data that you collected in your class or in your research lab. Explore our other data sets, compare, say, the human data to mouse data, or explore more of our human data. Explore that single cell transcriptomics data. You can do either combining our data with your data or analyzing our data alone. You don't need any permission from us in order to use our data for classroom or research use, but we of course love to hear about projects, so please let us know what you and your students are up to. Another approach is to practice reading scientific papers and comparing them to our data. You can find links to all of our scientific publications on our website, and those publications use the data that can be found on the site. And for students who are completing non-research projects about sharing science and health information, such as IB projects, our data and findings can be used to support outreach efforts as well. And this screen cap is from a video highlighting a high school student who used our data from a data set that we didn't highlight in today's webinar, the Allen Brain Observatory, for a project that she then took to the Society for Neuroscience annual meeting, a professional neuroscience conference. This video is linked from the lesson plan document, or you can find it on our website or on YouTube. So again, back to those learning goals, we hope that students will have taken away from this lesson some key open questions in neuroscience, some data types that are available to start addressing that, and start thinking about what questions intrigue them about neuroscience and how they can start asking and addressing them. So I am part of the communications department, and basically our role is helping put science into the hands of people who can use it: scientists, educators, general public, anyone. In addition to working with you, educators, we also have multimedia materials, some of which we point you and your students to during the lesson, videos, photos, stories. Under usual circumstances, we also offer live events for scientists to learn how to use our resources in their research, just like you're here today, learning how to use our resources in your classroom. While all of our events are virtual now, they're still happening. So education and educational users fit into this right alongside our research users. If you happen to also be interested in learning about our data for research, please keep an eye on our events page in addition to our education page to be learning about upcoming events. So my path to training and outreach, I started college thinking I would study animal behavior, specifically human-animal interactions related to climate change and shifting patterns of animal behavior as a result, and that I would be a biology major. So I thought I should learn about the brain in order to study animal and human behavior, and then I ended up loving it and going into neuroscience. I didn't even know that neuroscience was a field of its own until I got to college. I went to grad school also in neuroscience. I picked the program at UW because I liked that it was interdisciplinary, not just cognitive neuroscientists like me. I've always done research in humans, ironically, never studied animals. That I would be able to learn about a breadth of the field of neuroscience. In addition to my research in cognitive neuroscience, specifically functional connectivity in the brain, which is how different brain regions pass information back and forth and how they change as a result of learning a new skill, I ended up associated with some neural engineering projects because the technology I used for my research, which is called electrocorticography, has applications for prosthetics. In grad school, I always loved teaching and outreach, and that's what brought me to my current position, which I like to describe my job using this chart. On the y-axis, we have the Allen Institute for Brain Science and Cell Science. I also sometimes work with the Allen Institute for Immunology, but not as much because they haven't released their first set of public data yet. When I say scientists versus educators, I mean people who are primarily using our open resources for research versus primarily using the resources as teaching tools. Scientists are also educators. Educators are also scientists. Many people fall more strongly one way or the other, or in a particular application for our data, sit on more on one side of that than the other, but nobody's only one. And you are here down in the bottom right corner. We are talking about the Allen Institute for Brain Science in an educational application. And then training and outreach. When I say outreach, I usually mean helping scientists and educators learn about the resources we have available and figure out which ones are the best fit, and training to help people learn about the resources and actually use them. So that's where I fit into the communications department and getting all of the data and all of the findings that we have collected as an organization, all of that 8 petabytes of data into the hands of people who can really use it. So in summary, students can use our open online databases to conduct experiments virtually. They'll be exposed to data that they might not otherwise be able to collect or access. And while a virtual experiment cannot replace a live experiment, they can introduce a new type of data or serve as a bridge while you're learning at home. Again, you can find this lesson and more resources for educators at alleninstitute.org/learn. Have you used any resources in your class? Please let us know. Communications@alleninstitute.org if you use this. If you use anything else, please get in touch. And please join us for upcoming webinars. We are working on scheduling our next webinar now, and we will notify you when registration opens. So big thanks to the Allen Institute founder, Paul G. Allen, for his vision, encouragement, and support, and to the whole team of the Allen Institute. We have about 500 people. We truly do work in team science, and we couldn't do it without every single one. Well, we are just about at time here. So if you have any follow-up questions for us, we will set up a post in the BrainMap community forum where you can post your questions. You can see the answers to the questions. We'll share all of the answers we typed out. Or you can get in touch with us at communications@alleninstitute.org. Thank you so much for being here today. And we hope you have a wonderful rest of your day and a good semester this fall. Thank you.
Thank you, everyone.